Now, the virus being inside the cell has the potential for altering the host cell. If it's an envelope virus and you get a bunch of them leaving, all at once, there could be some damage. Because if they're leaving one at a time slowly, well, the the Golgi can maintain the plasma membrane. But if they're all leaving at once in a big rush, well, the Golgi and the can't keep up. That cell is going to be on the verge of splitting open. Any damage that can be done, any changes to a host cell or surrounding host cells due to a viral infection is referred to as cytopathic effects. So these cytopathic effects, it's a wide range. It can be anywhere from a slight change in metabolism to a mutation in the DNA genome of the host cell to complete alteration of a tissue. Great example of this is what's referred to as syncytia. Syncytia are where a group of virally infected cells all fuse together into one. One giant cell with multiple nuclei that are all trying to affect the running of the cell against each other. The syncytia can't last very long. They usually die off within a day or so. Great example of this, as you see here, is RSV, respiratory syncytial virus. You see here to the far side, these big giant cells with multiple nuclei. They're so big, they have trouble transporting in enough nutrients, having trouble getting enough oxygen throughout. And then you have the multiple nuclei all counter-signaling against each other. Sometimes you also get, as you see here, what are referred to as inclusion bodies. Instead of the nucleocapsids leaving, as soon as they're assembled, they'll clump together and form larger and larger clumps of viruses to be released later. These inclusion bodies will gum up the works. They will get bigger and bigger as they take up more and more volume within the cell. Another problem we see with viral infections is, it's not in and out, you're good to go. Sometimes you get what's referred to as a persistent infection. Viral DNA will actually insert into your host genome. So you get infected with a virus, that virus DNA then inserts and now becomes part of your DNA. They call this a provirus. The provirus looks exactly like your genome. Same A, G, C's, and T's are there. Every time your cell divides due to mitosis to replace, repair tissue damage or whatever, that provirus goes along with the daughter cells. Some point later in time, that provirus may start making new viruses. You'll get overwhelmed later. Think HIV. HIV, the DNA hides in our host genome cells, which is why our immune system can't find it for long, long periods of time. And then something will occur, and the provirus will start making copies, start making viral messenger RNA, which leads to the making of viral proteins, the making of viral virions, nucleocapsids, and next thing you know, your viral load in your blood starts to go up again a decade or two after you were infected. Sometimes you get what's referred to as a chronic latent state. You're constantly making the virus, but it's so low, your body really doesn't recognize it. Herpes, the gift that keeps on giving. We'll talk more about this when we talk the infection chapters, when we talk genital urinary infections. You're constantly, once you get the cold sores or the genital sores, you're constantly making the virus. It's constantly there, but it's at such low levels, well below the threshold, that your immune system needs to see to react to it. Same with the chickenpox. Chickenpox is one of those things that once you get it, you've got it for life. You have an immunity to it for life. Well, you also have a slight chance of having a chronic latent state where you're constantly making the virus. Same with measles. We see people get it. Most people get it. They mount an immune response. It's cleared. Some people get it, and it's always there. It's low levels, too low for their immune system to really get amped up about, to get activated for, but it's always there. The big worry, especially now in the day and in the age of cancer, is that some of the cancers are actually caused by viral infections. What happens? A virus infects a cell. It changes that cell. That change, it transforms the cell, makes it become something new. It's no longer a liver cell. It's no longer, an epithelial cell lining your large intestine. It's been transformed into a cancer cell. That's why they refer to it as transformation. These viruses that can cause that, they are grouped together and called oncoviruses. Onco means cancer. Oncology, study of cancers. Oncoviruses, cancer-causing viruses. They get in, they act as a provirus, and they insert into the host genome. Problem is that they have a tendency to insert into the genes responsible for regulating cell cycle. Can't regulate cell cycle, it means your cells are going to go through mitosis over and over and over. Or the cells lose the ability to control checkpoints. Remember, the checkpoints are how our cells say yes, no, whether or not they need to go through the next part of the cell cycle leading to mitosis. Ultimately, cancer is nothing more than the loss of control. Technically, that's the definition of cancer. Cells losing control of their replication process. So they start to grow and grow, increasing in numbers, not growing in size, but increasing in numbers. 1 becomes 2, 4, 8, blah, blah, blah. Next thing you know, you have a tumor. Here you see, look at this picture. You can see how it will work. These viruses insert into the host cell genome, interfering with normal processes, normal control mechanisms. So, retroviruses, RNA, the DNA traffics to the nucleus, becomes part of the host cell genome. Viral DNA is released, traffics straight to the nucleus, straight to being a provirus, and interferes.